Please come in. Please have a seat. Hi, good afternoon.
Hi, good afternoon.
Nice to see you. My name is Mariana Pop. I'm an associate professor in Pécs University. I teach molecular cell biology. I'm going to be your examiner today. I'm going to have a few general questions at the beginning and 2 biology and 2 chemistry questions. Okay, let's start with the general part. What is your name?
My name is Noam Schwartz.
May I see your ID card,
Yes,
please?
of course.
Thank you. Okay, thank you. What is your nationality?
Israel.
What is your date of birth?
It's the 27th of March, 1994.
Okay, which program are you applying for?
General medicine.
Okay, why is this choice, or why do you want to be a medical doctor?
Well, in my family, there is no any kind of medical related professionals. And in the high school, I was really interested in biology and all the scientific materials we have learned. So when I was 16, I decided to get more deeper insight into the science world and scientific world. So that's why I'm here.
Okay, sounds great. So why did you choose Pécs University? Have you heard about, or how have you heard about the university? Do you have any relatives or friends here who studies here?
Unfortunately, relatives I don't have here, but I have some Israeli friends who already graduated from here, and they had very nice opinion about this school and also about the students' life. They also mentioned that here it is much better to focus on studies because this is a smaller city, so everything is smaller but a little bit more familiar.
Yeah, and then we have a really a lot of students, international students from all around the world, so you will find a lot of friends here. What do you like to do in your free time? Do you have any hobbies or do you do any sports?
I like reading books. I do sport regularly, Pilates and yoga, and I play the piano, not very well, but I try to increase my talent in it.
How long do you play
It's been
piano?
6 years.
Okay, thank you. Have you ever read a book in English?
Yes, I like philosophy, so I read books in English, in philosophy mostly, but other topics as well.
Okay. Have you ever done any volunteer work?
Yes, back in Israel, I worked for an association who had the main task to help children there, mostly those who had some kind of economic problems. And we give aid, sometimes food, for example, or we do several kind of activities there.
Okay, great. Thank you for your answers, and let's start the scientific part. Let's start with the biology. My first question is the structure of the cell nucleus. So what are the parts of the nucleus?
Well, in the nucleus we can find the nucleolus, we can find the chromatin, and around the nucleus we can find the nuclear envelope.
What does it mean, chromatin? What does it consist of?
It consists of the nuclear material packed in a mass with proteins.
Okay, what are those proteins? What's their name?
Like the polymerase, for example, or enzymatic proteins.
But the structural proteins bound to the DNA, what is the name of those proteins? They bound to the DNA strand and form the chromatin structure.
Oh, the histone proteins.
Yeah, these are
yes,
the histone proteins, correct. Okay, what are the types of chromatin?
We have euchromatins and heterochromatins.
Okay, what's the difference between them?
The transcription and the activity in the transcription. And the euchromatin is under transcription, or it goes through the process, while the heterochromatin is not going to go through this kind of process.
So it's inactive, transcriptionally inactive. Okay, perfect. Do you know what types of processes take place in the nucleus?
Yes, for example, the DNA replication, the transcription, the DNA repair.
Processes, very good. Do you know any proteins involved in those processes which are basically found in the nucleus?
Yes. For example, the DNA or RNA polymerases, the enzymatic proteins which works in, for example, the DNA repair.
DNA repair proteins as well. Perfect. Where are they synthesized, those proteins?
In the cytoplasm of the cell.
Okay, and they're gonna get transported afterwards into the nucleus. Great. Okay, let's see your other question in biology. What do you know about the digestive system in humans? So what are the parts of this digestive system?
Well, the digestive system starts with our oral cavity and the teeth, and it goes through the esophagus and gets into the stomach, what we have eaten. And then we have the bowel system, first the duodenum and then the small intestine parts, and then we will have the large intestines and then the rectal and the anal canal.
Do you know the digestive enzymes found in this, any parts? Then can you mention any digestive enzyme involved in this process?
Yes, for example, amylase, which is an enzyme, or the pepsin.
Where does it found, the amylase?
Amylase is already found in the oral cavity as well, and later on in the duodenum from the...
What kind of macromolecule is going to be digested by the amylase?
The carbohydrates.
Okay, perfect. What is the role of the liver in this digestive process?
Well, the liver has a bunch of functions. One is that it stores glucose in the form of glycogen. It also stores proteins, and it produces bile.
What is the role of bile?
Well, the bile emulsifies the half-digested food, but in the lipid forms. So it's, but it does not contain enzymatic proteins inside.
Okay, perfect. Okay, what kind of enzymes are found in the pancreatic juice?
We have all kinds of enzymes that we can compare it with the macromolecules. So amylase that I have already mentioned. There is also lipase, which is for the lipids, the nucleus, which is for the nucleic acids as well.
Okay. What do you know about the absorption process? How does it happen? And which part of the digestive system?
The absorption goes through in the small intestines mostly. In the small intestines we have so-called villi, and these villi actually increases the surface of the intestines, so it helps the absorption and the process of it.
Okay, perfect. Do you know any diseases, human diseases, which are connected with this the abnormality of this digestive system?
Yes, one very famous is the lactose intolerance, for example, or the celiac diseases, or diarrhea.
Okay, perfect. Let's talk a bit about chemistry part. My first question is the structure of atoms. So what do you know about this general structure?
Yes, in the atoms we can find the protons. We can also find the neutrons and around the electrons.
So the atomic nucleus contains the protons and the neutrons, and then around this there are the electrons. Okay, what does it mean, atomic number?
The atomic number is a number that we can find in the periodic table. all of the elements have an atomic number, and it will tell us how many protons we can find in that element. And also the electrons.
Yeah, it's equals with the number of electrons. Okay. What does it mean, the mass number? What is the difference between the atomic number and the mass number?
The mass number is going to give us information about the element's mass, and we can calculate from each other the other one.
So this is the number of protons plus the mass number is the plus the number of neutrons. Okay. One important term in this topic, the isotopes. What does it mean? What is the definition of isotopes?
The isotopes means that we have an element which has a specific number of protons but different numbers of neutrons inside.
Do you know the types of the isotopes of the hydrogen atom, for example?
Yes, it has hydrogen 1, 2 and 3, which is the deuterium and the tritium.
Yeah, okay. Which one is radioactive, what do you think? Have you heard about this?
The third one.
The tritium, okay, that is radioactive and can be used for labeling different macromolecules. Okay, so what do you know about the localization of the electrons around this atomic nucleus?
Yes, actually the electrons can be found on shells, and these shells can be numbered, so we usually label it with N1, 2 and 3.
Which one is the closest to the atomic nucleus?
The N1.
Okay, okay. And then how do we name the electrons which are in the outermost shelf or layer?
These are the valence electrons.
Let's see an example. Let's pick an atom, let's say sodium atom. Could you please describe how many electrons does it have and how do they arrange on those shelves?
Yes, so the atomic number is 11. So we will have 11 electrons. And if we count it, we will have 3 shells, and on the third we will have the valence electron and number 1.
How many electrons can occupy those shells?
2.
The first one?
The first one is 2, then we will have 8 and then 18.
Okay, okay, great, thank you. Let's see another question in chemistry, your second one. What do you know about the structure of amino acids, general structure? Could you please show me the general structure of an amino acid?
Yes, of course.
Just name the parts of this.
I would usually draw with
Okay, so what are those parts? Can you name those
parts? Yes, one part is the amino part, one is the carboxylic part. We have a hydrogen atom and the central carbon atom, and then we will have the side chains, which is specific.
Okay. How many amino acids do we find in our cells?
20.
20 different. What is the difference between them?
The side chains.
The side chains. And what are the types of the amino acids?
We have aromatic, polar, apolar, acidic or basic.
So how do they bind together? Could you please show me another amino acid next to it and connect them?
Yes, of course. So we will have a water that goes out of this system, and then we will have a peptide bound in between each other.
Okay, perfect. How do we name the ends of a polypeptide chain?
The part where we have the amino part is the N part, and the other one is the C, and terminus and C terminus.
Okay, perfect. These are the ends of a polypeptide chain. Okay, perfect. So let's talk a bit about the structure of proteins. So what are the structural levels?
We have primary, secondary, tertiary, and quaternary.
Okay, so what does it mean, primary structure?
It's actually the amino acids that are combined into each other.
Okay, bind together by
the peptide bonds.
Peptide bond. Okay. How about the secondary structure?
We have two alternatives in there. We have one alpha helix and beta sheet.
What stabilizes this structure? What kind of bonds stabilize
Secondary bonds, usually the hydrogen bonds.
Hydrogen bonds, okay. How about the tertiary structure?
In the tertiary structures, we have, for example, van der Waals or disulfide bonds in between each other.
What does it depend on? Which
Depends
bonds
on the
stabilize?
side chain.
Yeah, so the properties of the side chain determine the bond between those parts of the polypeptide chain. Perfect. How about the quaternary structure?
In the quaternary, we have more tertiary proteins that are combined into each other.
Yeah, so more than one polypeptide chain bind together. Do you know any example?
Like hemoglobin?
The hemoglobin is a good example for the quaternary structure. Not all proteins have the quaternary structure, but hemoglobin is a good example. Okay. What could be the function, those proteins in the cell? Do you know any functional categories?
For example, enzymatic function could be mentioned, or transport function.
Transport proteins or enzymes, okay? Okay, or structural proteins?
Yes, structural proteins, yes.
Okay, perfect. Thank you for your answers and thank you for coming to this entrance exam. A committee will decide based on the result of the written exam and then this oral exam, and then they will send an email about their decision whether you are accepted or not. Thank you.
Thank you.